because you. So I'm talking about Abdul Fattah Yusuf Mohammed Hussein Mohammed. Just one. Okay. Akmal Isma. Al-Ghazali. Al-Umar Ghazal. Ghazal. Not here. Fawad Abdullah. Feroz Mehbaz. Hamid Abdulaziz. Izu Kareo. Ike Yaku. Chinazae Pere. Itkin Alik. Joseph Diana. Itamura Navia. Ma Shinosuke. Marul Amit. Nasser Munir. Rahman Rumana. Said Arwa. Segal An. Shehata Yusuf. Shehata Yusuf. Ugwanga Teopolina. Wang Mengia. Abdel Monen Yusuf. Akwaye Quadro. Aidu Ampofo. Aidu Ampofo. Not here. Ayaber Hala. Ayaser Rabi. Al Mohammed Al Elabi. Al Saman Abdallah. Amro Habiba. Shabda Jaibiai. Farah Rama. Kantman Patrice. Young Viaye. Kamire Yasmin. Katz Adiel. Levi Gali. Mahbub Mariam. Mohammadi Abu Taleb. Murata Hatsune. Yes. Naiga Kevin. Nishimura Shikibu. Yes. Okeke Indrani. Ater Krish. Reyuvergenov Asadbek. San Su Chan. Shamir Ria. Yes. Unuma Misaki. Khan Ivan Yakov. Sama Bashar. And everybody could hear his or her name. Okay. So please remind me what we did last occasion. My recall is that we finished the ovary and the uterine tube. And what about the uterus?
Yes.
Also. Okay. So was last time, and we finished all of these. Okay. Then we are all right. Today we deal with the pregnant uterus and also the placenta. So let's start with the number 78 section, which is the pregnant uterus. We need to refer back to the non-pregnant uterus section. Hopefully you will remember. So, yes, this is the slide. I think we should start with some drawings. So, we need to assume now that fertilization has occurred, which usually occurs in the ampullary region of the uterine tube, as we discussed. Then zygote is formed, a series of meiotic divisions start, like here, and this process is called cleavage, and the individual cells which are formed during cleavage are called how do we call the individual cells which are formed during cleavage? No, morula is the 16 cell stage status. It is not an individual cell. Blastomere, yeah, somebody learned. And then the morula, as somebody else has mentioned. And what is the next stage, which has a separate name after the morula? Blastocyst, which has how many different parts?
Two.
No, 3.
Oh.
What are those? Trophoblast. Yes. And the third part? No, not yet. Parts of the blastocyst. No, because it will happen later. We have only trophoblast. No cytotrophoblast and cystiotrophoblast yet. It has been mentioned. So we have trophoblast and embryoblast, and what is the other named part of the blastocyst? It happens later. Epiblast appears later.
Are you talking about the cavity?
Yes, which has a name, blastocyst. Oh, everybody passed embryology. Somehow. Unbelievable. The blastocyst arrives at the uterine cavity and starts its embedding process. And approximately how many days are required between fertilization and the start of the embedding?
2 weeks?
No.
10 days?
No. This is not a lottery. About 1 week, so 7 days. Do you agree? Thank you.
Is that the gastrulation?
What about gastrulation?
Is the week in the gastrulation?
Which week?
The first week. Or is the second week?
Oh, Jesus. Gastrulation happens during the 3rd week.
Third week, okay.
because that's the formation of the three-laminar germ. This three-laminar weak 3. It's so difficult.
Thank you. Forgot it. Thank you.
What is the stage of this, what you can see? How would you call it?
Implantation.
Okay, implantation has completed. No, I mean, how do you call this structure? Especially this one, which is in blue and yellow. Bilaminar germ disc. When does this structure form? Yes. Bilaminar germ disc V2. Trilaminar germ disc V3, because embryology is also complicated. Okay. Here is the amniotic cavity, which starts to form how? It's already here, but how does it start to appear? Cavities appear in the epiglottis, and those cavities will unite, and the united single cavity is called the amniotic cavity. And what is the origin of this epiblast and hypoblast? The embryoblast is the origin for both. Okay. And this one here, this cavity, this is the primitive yolk sac. Primitive yolk sac. How do we call these cells here? This single cell row. It's not written there. That's why I'm asking. So these cells here.
Is it the trophoblast?
No. No. No more embryoblast. Embryoblast already differentiated to epiblast and hypoblast.
Trophoblast. I said trophoblast.
No. Amnioblast. Amnioblast. So many blasts. Amnioblast cells here. Yes. And by this stage, this is from your book. But seemingly you cannot remember. This is the Hungarian version of the same book what you have in English. Better not to say anything. So trophoblast already differentiated into cytotrophoblast, which is this territory, this light gray, and this darker gray is the syncytiotrophoblast. What is the difference between the two?
They have a common liquid. The syncytotrophoblast has no membranes between cells. The nucleus are just floating inside.
Yeah, so no cell boundary in the syncytiotrophoblast. That's why it is called syncytium. So it's a multinucleated structure. Yeah. And what do you think about this columnar cell row here? And
Endometrium?
what's this?
What? Endometrium?
Well, the endometrium is not just a single cell row. Any idea?
Can you show what you're asking again?
This columnar cell row here and also here. What do you think? It is not written, that's why I'm asking. Yes, surface epithelium of what?
Of the uterus.
Of the uterus. Yes. Okay. Now, what are these red things here? Blood. Of whom?
The mother.
Of the mother. And this is where the endometrium or already closed above the implant, okay? And it is called fibrin coagulum in English as well. It is seen as a homogeneously eosinophilically stained substance in the section, which is kind of protein-rich structure derived from the blood of the mother, and it is protein-rich, so that's why eosinophilic. So obviously there was bleeding here during the process of implantation. My question is, what is your question first?
Is that going to be the placenta?
Pardon?
The fibrinocoagulum, is that going to be the placenta? Is it the beginning?
Nothing to do with the placenta, no placenta as yet.
Okay.
It will appear much later.
Okay.
I forgot my question. My question is, how is it possible that this structure is embedded into the endometrium? What breaks down what? Enzymes breaks down what? What? The endometrium. And who creates the enzymes which are used for this purpose? Which part of the blastocyst? The C-cystia trophoblast, yes. Okay. So that's why bleeding appears, because these enzymes disrupt the wall of maternal blood vessels inside the endometrium. Okay. Now, so let's look at this one as the last slide for now, showing the stage at the end of the second month approximately, and also at the end of the third month, also from your book, okay? So don't worry too much unless you don't have the book. We have the embryo here, which is located in this cavity called amniotic cavity. And the wall of the amniotic cavity is here, called amnion, which consists of an epithelial layer and a connective tissue layer. Okay? Outside the wall of the amnion, we have another cavity. That's the chorionic cavity, or also called, the other name, for chorionic cavity. It's not written, that's why I'm asking. Extra-embryonic cavity, that's the same. And we have a third cavity here, which is the uterine cavity. Okay. And we have some kind of light brown color here. I'm talking about this color. Everywhere where you can see this color, all those territories collectively decidua. And my question is, what is decidua?
What forms the
it is too general to be true, but decidua, part of the decidua has something to do with the placenta, yes, but what you are saying is not true because too general.
It's the lateral mesoderm.
Nothing to do with the lateral plate mesoderm. It's already disappeared because
No,
embryo is already here, so such structure as lateral plate mesoderm is no longer.
It's not derived from.
No. The light brown is decidua, as somebody correctly said. And then I ask, please define the term decidua. Yeah.
Is it modified intonation?
Modified. What do you mean?
During the pregnancy, the life.
Almost okay. So it is, you are saying basically that it is the endometrium of the pregnant uterus. Almost okay. A slight refinement is needed. No. So.
Does it provide any form of like a barrier between?
No, I didn't ask for the functions. So the answer is decidua is nothing else than the functional layer of the endometrium of the pregnant uterus. The functional layer of the endometrium of the pregnant uterus. That's the decidua. Okay? And decidua has several parts. Underneath the embryo here, so this is the territory where implantation started. This part of the decidua is called decidua basalis. Decidua basalis. Then above the implant, above the embryo, the territory where the endometrium closed above the implant, like a cap, this territory, therefore called decidua capsularis, from the word cap. Okay? Decidua capsularis. It is like a cap above the implanted structure here. And the third part is here, called decidua parietalis, written here. Decidua parietalis, this part and also this part. So these are the parts of the decidua which has no direct relationship to the embryo. Okay? Decidua parietalis. And it is not written in this drawing, but we can distinguish even a fourth part. Which is, because this is just a drawing showing the structures in a sectional view. In this sectional view, this structure is just 2 points. This point here and that point there. In 3 dimensions, it is a closed curve. And this is called decidua marginalis. Decidua marginalis. Actually, decidua marginalis is nothing else than the curve along which the previously mentioned 3 parts of the decidua meet. Okay, so this is where you can go to decidua parietalis, you can go to decidua basalis, and you can go to decidua capsularis as well, immediately from the decidua marginalis, because this is the meeting territory, which is a closed curve in 3 dimensions. In the drawing, just 2 points here and there, because this is just a sectional view. Okay, decidua marginalis. You will need to recognize these in the section as well, just to make sure you understand the importance. Okay, and let's look at this later later stage. One major difference is that in this drawing you cannot see the chorionic cavity anymore. It disappeared. And we have amniotic cavity within which the embryo is located, and we have a smaller but still existing uterine cavity. Smaller because the embryo has grown, and that's why the amniotic cavity also has grown. So what happened to the chorionic cavity? Here is the chorionic cavity, okay, between the wall of the amniotic cavity and the decidua. Now, this chorionic cavity disappears because the amniotic cavity is expanding, and the wall of the amniotic cavity, this, gets closer and closer to the decidua, and eventually we'll reach it and we'll fuse with it. That's why the chorionic cavity will disappear. Well, in most pregnancies, not in all, but in most pregnancies, the chorionic cavity disappears at later phases. All right. So that's enough for now as an introduction. So what can we recognize here? So first of all, we have a cavity here, which is seemingly open, but that's an artifact. This is the chorionic cavity. And you cannot see the embryo inside, and you cannot see the wall of the amniotic cavity either, because the embryo has been removed together with the wall of the amniotic cavity, and because of the removal, the chorionic cavity is open, as you can see. And because all these structures, including the substantially big amniotic cavity together with the amniotic fluid and embryo had been removed, that's why this cavity is quite elongated and not very spherical, so that the shape is not realistic either. Okay? And here is the wall of the chorionic cavity. This is the wall, also here on the other side. And you can call this as chorionic plate. Chorionic plate, the wall of the chorionic cavity. Now, this territory here, also here, this territory, decidua basalis, here and also here. The proof is, if I magnify, we can see here characteristic decidual cells like this, or like this, or like this. Well, it is partly here, this territory, but partly also here, this territory. So if I magnify this territory or this territory, it doesn't matter, because in both territories we can see these decidua cells, which are only found in the decidua. So that's why the presence of these cells is a proof that we are in the decidua. These are pale but basophilic cells. Cytoplasm is showing pale basophilia, and the cells are bigger than the nearby other cells. Remember, these are derived from the pseudo-decidual cells, what we already described. And pseudo-decidual cells are derived from what? From stromal cells, which are cells of the stromal cells are what kind of cells? Hmm? Embryo.
Structural cells.
Jesus, no.
Structural cells.
Hmm?
Structural cells. Structural cells.
Connective tissue cells of the endometrium. That's the stromal cell. And they undergo a transformation and become pseudodecidual cells in the secretory phase. And these pseudodecidual cells will become true decidual cells in case of implantation. And this is what we can see here, decidual cells. This is the true decidual cells without the pseudo prefix. Yes?
But you said that the decidual cells are functional units.
They have a function to accumulate lots of glycogen and lipids which serve as nutrient for the implant. That's the function.
So it's not stromal?
It used to be a stromal cell, but underwent a kind of transformation. How do we call that transformation? Epithelioid transformation. And thereby it has become pseudodecidual cells and later become decidual cells.
Okay, thank you.
Yeah. So, decidual basalis here and also here. Now, this part, what I'm showing now, this part, this is the decidua capsularis. Decidua capsularis. That this is part of the decidua is again based on the fact that if I enlarge, we can see these decidua cells. Can you see? Okay. This kind of fold represents the decidua marginalis, one point, decidua marginalis, and beyond that point, this territory here, decidua horizontales. Again, if I magnify, you can see the decidua cells.
They contain less basophilic color, no?
Yeah, they look like less basophilic a little bit, but yeah.
In the decidual capsularis, you see there's like a thin layer that looks like smooth muscle. What is that like?
Smooth muscle is not here.
What is that like pink layer?
This?
Yeah.
Okay, this is fibrinogen, kind of fibrin coagulum, derived from the maternal blood. Okay, now this territory here, below the decidual basalis, this is myometrium. Myometrium. You can see this is artificially cut here. So the outer part of the myometrium and also the perimetrium are missing from the section. If I magnify anywhere here, you can see extremely elongated smooth muscle cells. Can you see how long this smooth muscle cell is? The longest smooth muscle cells are in the wall of the pregnant uterus in the myometrium. This is an early pregnancy, so these smooth muscle cells will elongate even further later. Okay, so we
Looks a
can...
bit like skeletal muscle.
Hmm?
Looks a little bit like skeletal muscle.
No, it doesn't. I disagree. Why are you saying this? I have no
Because
idea.
it's like elongated and the nucleus is like ellipsoid shape.
But in a striated muscle fiber or skeletal muscle fiber, we have elongated nuclei, yes, but many and only at the periphery. And this is not the case here.
I know it's not. I just said it looks like.
Okay.
I should keep that to myself.
I don't want to argue too much, but I would suggest not to refer to striated muscle in relation to this section during the exam because you create a quite big problem.
Okay.
So if during the exam basic tissue types are not recognized, that immediately creates a very serious problem. And in the uterus, in the wall, there is no striated muscle. You know, this kind of mix-up will initiate further questions about the muscle.
Yeah. It's just a comment I shouldn't have said.
Probably you are right.
Yeah.
Shouldn't. Okay. Now let's come back to the chorionic plate and look at the sublayers. So this is the cavity here. Content was removed. So actually this widest layer of this chorionic plate is an embryonic connective tissue. You can call it extra embryonic mesoderm as well. Extra embryonic mesoderm. And because this is a relatively undifferentiated embryological tissue, connective tissue, you can see these elongated mesenchymal cells here, which have processes like this, or like this, or like this, or like this. So these elongated cells are the mesenchymal cells. In addition, in this extraembryonic mesoderm, you may also call it chorionic mesoderm, by the way, referring to the fact that this is in the whole of the chorionic cavity. So, in addition to mesenchymal cells, you can see these roundish or oval, slightly oval-shaped cells with eosinophilic cytoplasm. These are kind of connective tissue macrophages which belong to the embryo. And these macrophages have a special Aussos name. They are called Hofbauer cells, capital H and double F inside. Hofbauer cells. Hofbauer cells. Plus the Hofbauer cells. Hofbauer cells. And something else. We also have blood vessels like this. These blood vessels contain embryonic blood. And the proof is that this is embryonic blood and not maternal blood, that you can see here in the lumen a nucleated red blood cell, actually two nucleated red blood cells are here. Because at this age, embryonic phase, early fertile phase, the blood contains still nucleated red blood cells. Even in adults, as red blood cells mature, up to a certain phase, up to the normoblast phase, we also learned this, by the way, taught it. that red blood cells are nucleated and normoblasts lose their nuclei. So this is embryonic red blood cell.
Until which stage it's still nucleated?
As I said, normoblast is the stage when the nucleus is lost during erythropoiesis. But at this age, at this embryonic or early fetal age, red blood cells don't lose their nuclei at any stage. What I refer to is in the adult. Okay, so we have external mesoderm or chorionic mesoderm. Next layer is this cuboidal cell row, a single cuboidal cell row here. This is the cytotrophoblast. And outer to this cytotrophoblast, this territory where you can see these much darker nuclei, this is the syncytiotrophoblast. Syncytiotrophoblast. Okay. So external mesoderm, cytotrophoblast, syncytiotrophoblast. Yes.
The whole black.
No black.
Can you zoom in again?
This is not black, still blue.
Okay, this is the syncytiotrophoblast.
It is coming. We just talked about the chorionic plate yet, but we haven't reached this stage. But it is coming. Please be patient a little bit. Okay, the next thing is that you can see outgrowths from this chorionic plate, like here, or like here, or quite tiny outgrowths, like this one, or like that one. Now, these kind of structures are called stem villi. This is a stem villus here, stem villus, a kind of outgrowth from the chorionic plate. And then these stem villi branch like a tree is branching as it is growing out from the soil. The soil is the chorionic plate now, and this is the trunk of the tree, the stem villus. And the branches of the stem villi are here in this territory and also in this territory. And you can see them as irregularly shaped structures, which doesn't seem to be connected to the chorionic plate, but they are. The only thing that we cannot see the connection because it is not in the plane of sectioning. But all these structures are actually branches of several stem villi. And these villi, these branches, are surrounded by something which is called intervillus space. This space among these villi is called intervillus space because they are among the chorionic villi. These villi are called chorionic villi, nothing to do with intestinal villi. I'm sorry, this is again not a very good name, but yeah. This intervillus space is not empty but filled up by maternal blood. And you can see red blood cells which are non-nucleated in this intervillus space, meaning that it cannot be the blood of the embryo, it is the blood of the mother. So in the intervillus space we have maternal blood. And maternal blood actually surrounds these villi here, these branches. These branches are therefore maybe called as floating villi, or may also be called as free villi. Floating or free villi. Now, some of these branches of the chorionic villus system grow as far as they reach the basal plate or decidobasalis, like here, and fuse with it. So such a villus is fusing with the decidobasalis. It's a continuous structure here. Therefore, such a villus which is fusing with the decidua is called anchoring villus. Anchoring villus. So we can talk about stem villi, floating villi, and anchoring villi. All those which are directly connected to the chorionic plate, like here, or like here, or here, or here. This is tiny. All the imaginations. Yes. Fibrinogen, we already discussed. Fibrinocoagulum or fibrinogen. Okay, now we can categorize the chorionic villi by using another other system. So the previous category system of chorionic villi was based on the geometric relationship of the given villus to the chorionic plate and to the basal plate and to the intervillus space. Now this other system is based on developmental phases of the chorionic villi. So I would like to show you a drawing from your official embryology book. So the most primitive chorionic villi appear at the beginning of the 3rd week. And they are called primary villi. And the primary villus consists of syncytiotrophoblast and cytotrophoblast only, nothing else. Internally we have the cytotrophoblast, peripherally we have the syncytiotrophoblast. This is the primary villus. Appears at the beginning of the 3rd week. A few days later, into the cytotrophoblast, extraembryonic mesoderm is growing. So once extraembryonic mesoderm appears in the center, that's already a secondary villus. A few days later, embryonic capillaries appear in this extraembryonic mesoderm. When blood vessels appear, it's already a tertiary villus. The first tertiary villi appear at the end of the 3rd week. So this whole process is just 1 week. Of course, after week 3, further and further villi appear. I'm just talking about when the first appear. That's the 3rd week. Yeah.
How do the embryo blood cells bind to oxygen if they have nucleus?
How oxygen gets to embryonic blood?
No, I get how it gets to the blood, but how do the red blood cells of the embryo that have a nucleus, how do they bind to oxygen?
Well, hemoglobin is not, hemoglobin is in the cytosol. So I don't understand the problem. nucleus is there, but it doesn't disturb oxygen take-up.
So red blood cells can bind to oxygen with the nucleus as well?
Independently whether the nucleus is there or not. Nucleus is not needed for this process. I don't know how much hemoglobin is present at this stage, but actually it is not needed either at this stage because not needed as much. I suppose hemoglobin appears in increasing concentrations gradually as the oxygen demand is growing because the whole embryo is growing. But don't go into details now because it is going far. So, for example, here I can show you a primary villus. It is so tangentially sectioned that actually it looks like just consists of cytotrophoblast because in the plane of sectioning we cannot see any syncytial trophoblast. But this is also a primary villus seemingly. But if you look at, let's say this villus, which is otherwise a floating villus, inside you can see extraembryonic mesoderm, and within the extraembryonic mesoderm you can see embryonic blood vessels. So this is already a tertiary villus. Okay. All right. Please look at the system of chorionic villi. Here, between the chorionic plate and the basal plate, in this territory, and compare the chorionic villus system here, which is between the chorionic plate and the decidual capsularis here. This is much less developed system, less branching, and the number of chorionic villi is also lower in this territory compared to this territory. Based on this observation, we can define two subsets of chorionic villi and we can name them. So this more developed subset, which is between the chorionic plate and the basal plate, this is the chorion frondosum. Chorion frondosum. More branchy, more developed part of the chorionic villus system. Chorion frondosum. And this other part, which is less developed and eventually will disappear without function, this is the chorion leve. Chorion leve. Chorion leve. Which means smooth chorion in Latin. Well, it is not smooth because it is also branching, but I would say smoother relative to the chorion frondosum, okay? So maybe this helps to memorize. Yeah.
So you believe both the primary and tertiary, but can you find any secondary villi?
You may find it. Look at this. We have external mesoderm, but we cannot see blood vessel. That's it. That's another story whether we cannot see the blood vessel because no blood vessel yet. But the other possibility, we cannot see the blood vessel because it is not in the plane of sectioning. We cannot decide. But you should know in theory what we call as secondary chorionic villus. Okay, once we are here, I just should refer to this. We are maybe a better example somewhere. Look at this cell. What's this? This is Hochbauer cell. So these Hochbauer cells are also here in the extraembryonic mesoderm of the villus. Okay? And of course these blood vessels inside the core of the villi, these blood vessels are connected with the blood vessels inside the chorionic plate. Okay? These blood vessels branch and grow into the chorionic villi. Okay. Well, it doesn't indicate. Obviously, Hochbauer cells can only be in secondary and tertiary villi because only those contain extraembryonic mesoderm where Hochbauer cells are found. So, in primary villus, you cannot find Hochbauer cell. But
So it eliminates just the possibility of it being primary villus.
in a primary villus, you cannot find Hochbauer cell because in a primary villus there is no extraembryonic mesoderm.
Sir, what are they derived from and what is their function? Is it the third?
That's a macrophage, so it's phagocytosis, major function. Yes.
The difference between the chorion leve and the frondosome is that the frondosome connects to the basal vestigial and the leve connects to the parietal.
No, capsular.
Oh, to the capsule.
But this is one difference. The other difference is that the chorion frondosome is more branchy and more developed.
And the leve more smooth. Kind of.
It is less branchy, less developed. So if one of the two things is more developed and more branchy, then the other one is less branchy and less developed. So I'm sorry, but
No, I agree with you.
okay, maybe that's my fault. So I thought it was obvious from this kind of definition. But yeah. So in this section, it is difficult to tell exactly the age of the pregnancy here, but it is early pregnancy, so probably not older than 3 months old. And at this stage, we still cannot talk about placenta. So I cannot show you the placenta because it is not yet developed fully. But I can show you, rather than chatting, if you pay attention now, we can avoid some further questions, maybe not. Because I'm just talking about the fact that no placenta is here yet, but we can point to structures from which the placenta will later develop. And in this context, the placenta has 2 major parts, fetal part and maternal part. Now, the fetal part of the placenta is developing from the chorion frondosome. So, chorion frondosome will give rise to the fetal part of the placenta later. And the maternal part of the placenta will develop from the basal plate later. Okay, so we can point to the structures which later give rise to the placenta, but it is not yet placenta. Now, so let's open the placenta section, 79, which is the so-called full-term placenta. This term tells that this is the fully matured placenta, how it looks like just before delivery. So we are going to look at the full-term placenta. Oh, I should have shown something else in the previous one, so I'm sorry, I need to go back just for a minute. It's already closed, I don't know why. Didn't close it at all. So, I should show something in the endometrium, actually two things. So we go back to the pregnant uterus section, just for a minute, and look at the basal plate. Here in the basal plate, first of all, you can see structures like this. This is a gland, an enlarged uterine gland. So these pale columnar cells are the secretory cells of the uterine gland. And the reason why these glands get so dilated is that when the anchoring villi reach the basal plate and fuse with it, the syncytiotrophoblast, okay, releases those proteolytic enzymes which disrupt the endometrium, and those proteolytic enzymes disrupt the glands as well. So obviously these glands are exocrine glands and release the secretum to the surface of the endometrium. But if this apical part of the gland has been degraded, then it closes the route for the secretum towards the cavity. And therefore the secretory cells keep on producing the secretum, which cannot leave. Therefore the secretory unit is expanding. So that's why you can see this dilated uterine gland here. Of course, these uterine glands produce glycogen and lipid, as we discussed, which serve additional nutrients for the embryo. This is also a gland here. Now, in addition, you could ask the origin of this maternal blood which is in the intervillous space. They are primarily from the spiral arteries of the endometrium, whose wall gets disrupted again by the enzymes produced by the syncytiotrophoblast of anchoring villi. Here you can see some lacunae. One lacuna is here. Dilated blood vessels, which are in the upper part of the endometrium. And so it contains maternal blood. And you can see this fibrinogen, this reddish substance, especially in territories where anchoring villi reach the endometrium. Because at those locations, the wall of blood vessels within the endometrium gets disrupted. And maternal blood is flowing out, and the fibrin content of maternal blood gives this kind of appearance. So there you can see this reddish marking, that's the sign of bleeding from blood vessels. Okay, now it's time to go to the placenta. So the placenta is a disc-shaped structure. You cannot see the full placenta, and you can see here an artificially cut edge, which is showing that in this region the placenta is thicker than at the periphery. That corresponds to the disc shape I mentioned. So this is the peripheral part. And the other thing which proves that we are here in the peripheral part of the placenta is this triangular shaped structure, which is a marginal vein. Marginal vein. So this is a vein. You can see if I enlarge that so the internal lining is given by endothelial cells. It's a marginal vein which collects the venous blood from the placenta, and it contains maternal blood. Okay? And it runs around the placenta, so it's a kind of circular oval-shaped vessel which is seen in sectional view. We should be able to see the other one here on the other side of the placenta, but the other side is just not here. Now, this territory here is the amniotic cavity. Chorionic cavity has disappeared because the wall of the chorionic cavity and the wall of the amniotic cavity have been fused. As a consequence, this is the fertile side, and this is the maternal side. Another proof here that here, this bluish territory, what I'm showing here, this bluish territory here, this is the basa plate. Because if I enlarge, you can see the deciduous cells. Okay? This is the basa plate. Or I can say now that this is the maternal part of the placenta. Okay? Maternal part of the placenta. Here. The chorionic plate is here, but the chorionic plate already fused with the wall of the amniotic cavity. And because of that, here on the other side, we can see a simple cuboidal epithelium, which is the amniotic epithelium, facing the amniotic cavity, amniotic epithelium. Below we have extraembryonic mesoderm, which used to belong to the wall of the amniotic cavity. Of course, we cannot tell exactly where the border is between the extraembryonic mesoderm of the amnion and the extraembryonic mesoderm of the wall of the late chorionic cavity, but somewhere these extraembryonic mesoderms have been fused. And within this extraembryonic mesoderm, we have 5 big blood vessels, which are branches of the umbilical vessels. Therefore, inside, fertile blood is flowing. So these are branches of umbilical vessels. If you look into the lumen, you can see non-nucleated red blood cells, because we are just before delivery. By this time, fertile blood contains non-nucleated red blood cells. This is already a lymphocyte, it is not a nucleus of a red blood cell, okay? So we have amniotic epithelium, we have extraembryonic mesoderm, and on the other side, other side, if I can find a good territory. Yeah, here. So here this is extrameric mesoderm, but here you can see cytotrophoblast, these paler nuclei belong to cytotrophoblast, and these darker nuclei, outer, that's the syncytiotrophoblast. Okay? So layers include amniotic epithelium, simple cuboidal epithelium, extrameric mesoderm with branches of umbilical vessels and Hofbauer cells and mesenheimer cells. Then cytotrophoblast and syncytiotrophoblast. And then we reach the intervillous space where maternal blood is found, which again contains non-nucleated red blood cells. So this is already maternal blood here. And you can see a very well developed system of chorionic villi. So this is what is derived from the chorion frondosum, but at this stage, this is already called the fertile part of the placenta. This chorionic villus system is the fertile part of the placenta. Okay? Yes?
In the middle, these are the free villi.
Okay, villi are coming in detail now. So look at this structure here. I think you are referring to this.
Yes, exactly.
Now, this is an anchoring villus. Because if you look at this part here, it reaches the basal plate and fused with it. So this is an anchoring villus here. These ones here are floating villi, surrounded by maternal blood. And look at this structure here. You may think that this is also an anchoring villus, but it is not. It is not. This is not a structure which is growing towards the basal plate, but this is a structure which is growing out of the basal plate. This is a placental septum, which is growing towards the chorionic plate, but never reaches it. And the difference between this anchoring villus and this placental septum is that in the placental septum we have decidual cells. In the anchoring villus, we don't have decidual cells. Instead, we have these elongated cell nuclei, which are actually primitive embryonic or fertile cells, used to be mesenheimer cells. But no decidual cell, that's the point. So that's the base of distinction, whether or not you can see decidual cell inside.
Right now, are you pointing at the anchoring villi?
Yes, this is an anchoring
Do
villus.
you see are these fibroblasts similar to the fibro?
Yeah, maybe fibroblasts are already here, which already differentiated from the mesenheimer cells. Because as you can see, lots of connective tissue fibers are already here, so we need to assume that probably fibroblasts already appeared. Okay. These placental septae divide the placenta into units. A placental unit is called cotyledo, starting with C, and it contains a Y, cotyledo. So between two placental septae, we have one cotyledo, and within one cotyledo, we have one or two with its with their branching chorionic villus system. Yeah. Now, let's magnify and look at the structure of these floating villi. So, one thing what you may recognize, that blood vessels inside the extraembryonic mesoderm of these villi tend to be at the periphery of the villus. Very rarely in the center. In tendency, they are rather in the periphery. This is one thing. The other thing, remember, inside these blood vessels within the villus, fetal blood is flowing. In the intervillus space, we have maternal blood. Normally, the two kinds of blood never mix. Never mix. Because there are several tissue layers in between. And all those tissue layers which separate these two blood compartments together are called placental barrier. Placental barrier. So placental barrier separates the fetal blood from maternal blood. That's why normally never mix. But material transport between the two blood compartments is possible in a controlled manner. Certain substances can pass through the barrier. So, what are the layers of this placental barrier? So, imagine that we are in, let's say, we are sitting on a carbon dioxide molecule, which is in the fetal blood. And obviously this carbon dioxide wants to go to the intervillus space to maternal blood. And then the lungs of the maternal lungs will get rid of the carbon dioxide produced by the fetus as well. So, the layers of the barrier starting from the fetal blood. First is the endothelium of the capillary. Then the basal lamina of the endothelium. Then there may be some extraembryonic mesoderm, maybe or maybe not. It is variable. Then we have another basal lamina which belongs to the cytotrophoblast. We cannot see it, too small, too thin. So basal lamina of the cytotrophoblast, then the cytotrophoblast, and finally we have the syncytial trophoblast. So these are the layers of the barrier. This is the so-called thick type of barrier because it includes extraembryonic mesoderm and two basal laminae. But sometimes these blood vessels get so close to the cytotrophoblast that the basal lamina of the endothelium and the basal lamina of the cytotrophoblast will meet and fuse, and the extraembryonic mesoderm which existed between the two disappears. So the barrier is thinning. This is how the thin type of barrier is formed. So in a thin type of barrier, we have just the endothelium, we have a common basal lamina, we have the cytotrophoblast, and we have the syncytial trophoblast. But even further, in a full-term placenta, even other changes may occur because in some occasions, the cytotrophoblast tend to disappear, like here. You can see the syncytial trophoblast, but this is a cytotrophoblast cell nucleus, but here I cannot see cytotrophoblast. At some other locations here, for example, the syncytial trophoblast is also missing. These are typical changes in the very late placenta stages. So these are kind of degradations, which creates a potential danger because the syncytial trophoblast coverage around the villi becomes discontinuous. And that creates a potential danger for the mother because in this context the syncytial trophoblast replaces the endothelium. Endothelium is always present in other territories, in vessels, in between the wall of the vessel and the blood. And it has many roles, but one role is to prevent blood from coagulation, because endothelium is negatively charged just like the proteins inside the blood. So electrostatic propulsion is there. And endothelium is not here, but the function of the endothelium is taken over by the syncytial trophoblast. But if syncytial trophoblast is missing, that creates a possibility for blood clotting, because the collagen here in the external mesoderm is positively charged. It will attract the negatively charged blood proteins, and it initiates or may initiate blood coagulation. So in rare cases, thrombus may appear in maternal blood at late stages of pregnancy. Yes?
Isn't there a basal lamina between them?
Basal lamina also contains collagen. It's part of the extraembryonic, part of the ECM, extracellular matrix.
So if the mother is on blood thinners, does this affect the flow of blood at all?
If mother takes blood thinner, obviously it will prevent blood from clotting, but I don't know whether it has side effects for the fetus. Okay, so we need to discuss here what can go through the placental barrier. So there are obviously harmful and useful substances as well which may go through. Useful substances include oxygen and carbon dioxide, and this way the placenta replaces the functioning lungs of the fetus, which are still not functioning. Also, nutrients from maternal blood may go through the barrier and may reach fetal blood. And these nutrients are already ready to be taken up by cells of the fetus. So the placenta this way replaces the digestive system of the fetus as well. Further, excretory products created by the fetal body may leave the fetal blood through the barrier and end up in maternal blood, and the excretory system of the mother will get rid of those excretory products produced actually by the fetus. So the placenta also replaces the excretory system of the fetus. Further, from maternal blood to fetal blood, immunoglobulins may go, which gives passive immunity, humoral immunity to the fetus. So, to a certain extent, it replaces the immune system of the fetus. So all these are useful substances. The most frequent Ig molecule is the IgG. Now, there are harmful substances which can also cross the barrier, like certain drugs, nicotine, alcohol, even pharmacons in medicines, which can harm the fetus. And also pathogens may cross the placental barrier from maternal blood to fetal blood. Zika virus, HIV virus, chickenpox virus.
Ethanol.
That's alcohol. I mean,
Yeah, it can pass.
yes. Mother shouldn't drink alcohol.
That's good.
Drink water. Okay. Coffee. Not good. So that's the responsibility of the mother, I'm sorry to say. So that's it. Thank you very much for your attention. See you on Friday. Please turn the computers on.